Hi everyone, thank you so much for joining today. My name is Rebecca Northeast and I'm the technical specialist for Proteintech. So thank you so much for joining our first ever Cancer Immunotherapy meeting. And we're really excited to get started. So just got a quick introduction slide. So the aim of this meeting is to provide a platform for early career researchers to present their work and helping to support them and gain visibility to aid their careers. And while we would really love for this event to be in person, one of the benefits of a virtual conference such as this is the lack of borders and visas, enabling us to bring together a speaker lineup and audience from across the globe. And one key aspect that is missing from in-person events is that all-important networking. And so I really want to encourage you all to reach out and connect with the speakers and each other on LinkedIn and Twitter. You can use the chat function in the webinar to be able to post your social details and to keep this conversation going, able to make these all-important connections. So this is the agenda for today. And now I will introduce and hand over to our session chair, Dr. Eduardo Bonavita. So Dr. Bonavita obtained his PhD in immunology at the Life Science Research Institute in Milan. During his PhD, he published landmark studies addressing the role of inflammation in modulating anti-tumor immunity, and he also discovered a new checkpoint receptor for natural killer cells. Since 2016, Dr. Bonavita works as a research scientist at the Cancer Research UK Manchester Institute in the laboratory of Dr. Santiago Zeleni. And here he is investigating the opposing role of inflammation in carcinogenesis and the contribution of innate immunity to supporting or restraining cancer progression. So over to you, Eduardo.
So thanks very much, Rebecca, for this kind introduction. And as Rebecca said, I will be chairing this session. So I'm very pleased to introduce our first speaker. So it's Dr. John Maher. So he will deliver the first keynote lecture of this meeting. Dr. John Maher is a clinical immunologist and is currently leading the CAR Mechanics Research Group at the King's College London. He played a fundamental role in early development of second-generation CAR technology, based on CD28, and this approach has achieved a great clinical impact in hematological malignancies. His research group is focused on the development of adoptive immunotherapy using CAR-engineered and gamma delta T cells, with the primary emphasis on solid tumor types. He's also the scientific founder and the chief scientific officer of a spin-out company named Lucid Bio. And in addition, he's a consultant immunologist within the King's Health Partners and Eastbourne Hospital. So today, Dr. Maher will be talking about CAR T-cell immunotherapy of solid tumor, learning from the clinic and the lab in parallel. So please, Dr. Maher, whenever you're ready.
Great. Thank you very much, Eduardo, for that kind introduction. And thanks also to the organizers at Proteintech for the opportunity to present some of our work on targeting of solid tumors with CAR T cells. And this slide just summarizes a number of disclosures. So I guess what I'm going to try to do in the next 20 or 25 minutes is to tell you two stories illustrating our experience with targeting of solid tumors using CAR-engineered T cells. The first of these stories relates to a clinical trial which we're running at the moment at Guy's Hospital, and it involves a CAR T cell product that we refer to as T4 immunotherapy. Now, T4 immunotherapy, you can see it illustrated here in cartoon format on this slide, and it consists of, on top, a second generation CAR that we call Ti-28 zeta, which is co-expressed with a chimeric cytokine receptor that we call 4-alpha beta. So let me tell you just a little bit about those two molecules and the thinking behind them. So the CAR that we're using here is a second generation CAR in which we have a CD28 co-stimulatory module placed upstream of a CD3 zeta activating unit. So by this means we deliver signal 1 and signal 2 to the T cells in a tumor target dependent manner. The slightly unusual thing about this CAR is the ectodomain shown by the red blob here. This is a chimeric polypeptide called T1E. And T1E was actually developed back in the Netherlands many years ago as a chimera between TGF-alpha and epidermal growth factor. And whilst both of the parent molecules are unique ligands for the epidermal growth factor receptor, when you make this artificial chimeric molecule, what you create is a promiscuous ERB ligand, which can bind to 8 out of 9 different ERB homo and heterodimers. So we thought this would be a useful targeting moiety with a view to trying to direct our T cells against essentially the entire ERB network. Now, we co-express this CAR, as I mentioned, with a chimeric cytokine receptor, which you see on the bottom there, called 4-alpha beta. And our thinking here relates to the fact that, of course, when we carry out research, we typically take blood from 20-something PhD students who are young, fit and healthy, have beautiful T cells, we achieve efficient gene transfer, lovely anti-tumor activity, and we publish our papers and move on. But of course, that doesn't reflect the reality of the patient population that you're dealing with, who tend to be elderly, they tend to be immune compromised because of disease burden and prior treatments, and often efficiency of gene transfer is much less impressive in this context. And so by engineering this chimeric cytokine receptor 4-alpha beta, what we have done is to create a unit which enables us to selectively expand the gene modified T cells using interleukin 4. Now, interleukin-4 is normally a very poor growth factor for T cells, but if you have this receptor in which you couple IL-4 receptor alpha ectodomain to the shared beta chain used by the receptors for interleukin-2 and 15, what you achieve using this strategy is you convert IL-4 into a selective growth factor for the genetically modified T cells. And so this is what we do in our manufacturing process. We activate the T cells from the patient, transduce with our retroviral vector, and then the only growth factor which we add to these T cells thereafter is interleukin-4. Now, the good side, I guess, of targeting the ERB network is it's pretty difficult to find a solid tumor in which this network is not expressed and indeed commonly is dysregulated. But the downside is the safety concern, because of course these receptors are expressed at lower levels in many normal tissues. And for this reason, we selected head and neck cancer as our target disease, because in contrast to most cancers, where metastatic disease is usually the cause of major morbidity and mortality, in head and neck cancer, it's this rather gruesome scenario that causes most of the trouble for these patients. It's locally advanced or locally recurrent tumor formation. And consequently, we reasoned that this provides a clinical disease model, as it were, in which you can assess and de-risk intratumoral delivery of a potentially toxic CAR T cell product. We also chose head and neck cancer for a number of other reasons. It's pretty common and it's getting commoner, and there is still a significant unmet need in terms of overall survival of patients. So we see a lot of patients who enter this type of scenario over the course of their treatment journey. And most of these, in fact, virtually all of these tumors express the target. They're virtually 100% positive for the epidermal growth factor receptor, and most of these tumors will co-express at least one of the other ERB family receptors. The other reason that we chose this disease relates to the really dismal survival prospects of these patients once they enter the arena of being untreatable using conventional medical approaches. The survival curve you see on this slide here was generated at our center at Guy's Hospital about 10 years ago or so, and it shows that these patients, once they have untreatable disease, begin to die of that cancer literally from day one. And by 30 weeks later, all of these patients will have died of head and neck cancer. And we found this to be a very important argument, I guess, to justify the ethics of what it was that we wanted to do in making the case to the regulatory authority, that we wanted to evaluate, as I say, this potentially toxic CAR T cell product delivered directly to the target lesion in these terminally ill patients. Now, the design of the clinical trial is pretty conventional when it comes to phase 1 or first-in-man evaluations of a new cancer medicine. So it's a dose escalation study in which we treat groups of 3 patients with initially a low dose level, and then we escalate through 5 different dose levels. In the context of this trial, our starting dose was 10 million CAR T cells, and we escalate up to a maximum deliverable dose of 1 billion CAR T cells. And these cells are formulated in a volume of between 1 and 4 milliliters. And as I mentioned earlier, they are injected as a fresh product directly into a target lesion, such as that demonstrated on the previous slide. Just before the COVID pandemic struck, we had opened a 6th cohort in this trial in which we preconditioned the patients with lymphodepleting chemotherapy. Now, in most CAR T-cell clinical trials, patients receive this type of preconditioning in order to create space for the infused CAR T-cells. But of course, our study is a little bit different in that we are delivering the cells directly to the tumor rather than into the bloodstream. So we decided to err on the side of caution in the first instance by omitting the use of lymphodepletion in the dose escalation phase of the study. And once we had demonstrated the safety of T4 through those 5 dose levels, we then proceeded to condition patients with Flu/Cy. That's a combination of fludarabine and cyclophosphamide, 2 drugs which are potently lympholytic and eliminate circulating T cells for a period of about 7 to 10 days. The primary endpoint of the study is to determine the safety of this, this new experimental medicine. But of course, we have a number of secondary endpoints which are focused around assessing efficacy and also a number of immunological parameters which may be perturbed in these patients. And the inclusion and exclusion criteria listed on the slide, again, are pretty conventional. This is standard for it's more or less any new cancer medicine evaluated in patients with locally advanced or locally recurrent head and neck cancer. Now, this is a busy slide and it summarizes where we stand in the trial at the moment in terms of patient recruitment. We have recruited a total of 18 patients to this trial, of whom 16 received treatment with T4 immunotherapy. Patient number 5 was a 50-year-old man who unfortunately died of his cancer in the 2-week period that we were manufacturing a batch of T4 for this individual. So clearly he was not treated. And the other patient who was not treated, patient number 12, was a young man from Eastern Europe. By the time he managed to get to the UK and acquire an NHS number, which is required to participate in these kinds of studies, he unfortunately had tumor which was invading major vasculature in the neck, and we couldn't safely inject CAR T cells into a lesion such as that. All of the other recruited patients were treated with the target dose of T4 immunotherapy. A couple of other points to take from this slide. Firstly, you will notice, if you look at the ages of these patients, that we have 2 octogenarians. So in other words, it is quite safe actually to inject CAR T cells directly into the tumor, especially if you're not giving lymphodepleting chemotherapy, to the extent that you can take elderly people and put them through this procedure quite safely. The other point to mention, shown on the right of this slide, relates to the prior treatment that these patients have had. And as you might expect, these are very heavily pre-treated patients with up to 8 lines of prior therapy before receiving T4 immunotherapy. The manufacturing process that we're using is a little bit different to that used in most CAR T cell clinical trials. We make these CAR T cells from a blood draw rather than from a leukapheresis. So we take about 120 milliliters of blood. It comes into our GMP manufacturing facility, and we carry out the procedure over a period of 2 weeks. You can see the growth of the cells on the left, and you can see the proportion of cells which express the CAR at the midpoint and the end of the manufacturing period shown on the right. And as I say, this is a 2-week manufacturing period that we're using. Notice also that the proportion of gene-modified cells increases when you compare midpoint and end of manufacture, and that reflects the selective advantage conferred by IL-4 upon the retrovirus modified T cells. From a safety point of view, T4 has proven to be very safe. We have not seen any dose-limiting toxicities, no cytokine storm, or indeed neurotoxicity. But we do see a consistent profile of side effects in these patients, whereby the target lesion becomes quite inflamed and painful, and this can persist sometimes for weeks. From an efficacy point of view, whilst we're not seeing anything like the same level of efficacy has been reported in hematological malignancies, there are a number of pointers to indicate that we are seeing an efficacy signal in this clinical trial. Firstly, in the first 13 patients who were evaluated for disease status, 9 of those patients achieved stable disease by RECIST criteria, where you essentially measure the volume of the tumor by scanning before and 6 weeks after treatment. One of these patients went on to enter another clinical trial in which he received a PD-1 checkpoint blocker, pembrolizumab, and an oncolytic virus, TVEC. And he went into complete remission. And that was more than 3 years ago. He's still in CR, having had a tumor the size of my fist growing on the side of his neck before he received T4 immunotherapy. The other indicator to efficacy relates to this survival curve based on intention to treat analysis. As I mentioned earlier, without treatment, all of these patients will die by 30 weeks, which is indicated by the black arrow on the x-axis of this plot. But if you look at the survival curve, you can see that it's been pushed to the right. I don't want to make too much of this because one commonly sees this kind of thing in clinical trials, but it's nonetheless encouraging. And on the right of this slide, this is the patient I just alluded to who went into CR. He's gone public with his story, and really quite a remarkable response, having had treatment refractory disease before entering into the study. I think this is my final slide on the trial, and it shows something else that we have done in one of the patients to monitor the migration of the CAR T cells. So what we've done here is to take a small aliquot of the CAR T cells, label them up with indium, and inject them into the target lesion. And what you can see on this scan here is all of this on the side of the face is cancer. And you can see that these T cells are sitting at the apex of that large tumor immediately after administration. By 24 hours later, they're still at the same location, but you can see a little bit of signal traveling across the oral cavity. And by 48 hours, you can see that most of the signal has actually migrated. We've only done this once, so this is kind of hand waving or speculative in terms of what may be going on here. But my suspicion is that this may reflect trafficking of the T cells through the highly disrupted lymphatic drainage of that tumor. This is a patient who would have had a neck dissection, so the normal lymphatic drainage of the tumor would have been disrupted by that surgical procedure. And he also had radiotherapy in addition, so very disrupted drainage profile of that lesion. So in the remainder of the talk, I'm going to move from the clinic back into the lab and tell you about a technology that we have been developing through Lucid Bio known as a parallel CAR. So much of the hoopla around CAR T stems from this second generation CAR technology in which you have a fusion receptor containing a single co-stimulatory module and a single activating domain. Now we know that if you provide 2 different flavors of co-stimulation to T cells, such as, for example, CD28 and 4-1BB co-stimulation, in physiological immune responses, they tend to synergize and you tend to get a more potent immune response. But this hasn't properly been capitalized upon in the context of CAR design, because when you make linear fusions, which contain 2 co-stim domains, such as a 3rd generation CAR, you don't really see much difference in terms of function compared to a 2nd gen CAR. And that caused us to speculate that the difficulty here is that you need these signaling units to be very close to the plasma membrane where they're physiologically located. And for this reason, we designed the parallel structure seen on the right of this slide, where you have a 2nd generation CAR with, for example, CD28 located in its natural membrane proximal position, and you pair that up with a 2nd receptor, which contains a different type of co-stimulatory module, such as 4-1BB. And by separating these out in two receptors, you can position both of the co-stimulatory domains at their natural location. Now, again, this is a somewhat busy slide, but essentially the point here is that we made a whole series of second generation, third generation, and parallel CARs targeted against a model target called the colony stimulating factor 1 receptor. This is a receptor that is expressed on a number of tumors in addition to tumor-associated macrophages. But the main reason we chose it actually in the initiation of this work is that there are conveniently two different natural ligands for this receptor, namely CSF1 itself and interleukin-34. So we could make all of these various CAR configurations in a kind of plug and play type fashion in order to evaluate the hypothesis that you need membrane proximity of the co-stim domains in order to get that synergy between CD28 and 4-1BB. And I'll mention one control here as well, where we have a first generation CAR and a dual co-stim second receptor. The idea here is by placing CD28 upstream of 4-1BB in this co-stimulatory receptor, we speculated that we may pay a price and not see as efficient co-stimulation as compared to the parallel CAR shown on the right here, where we have CD28 in this location and 4-1BB alongside it in a separate receptor system. Now, to compare these receptors, we carried out in vitro tumor restimulation experiments. And the idea is very simple. You simply take these engineered T cells and re-stimulate them on tumor monolayers which express the target antigen. And you keep re-stimulating the T cells in the absence of exogenous cytokine until the T cells become exhausted and can no longer proliferate or destroy the underlying tumor monolayer. And when you carry out that experiment, you find that all of the linear CARs, the second generation or third generation CARs, or indeed that control that I just pointed out, where you have a first gen CAR and a dual co-stim receptor, they all more or less behave in the same way. You can re-stimulate them through 2, 3, 4, 5 cycles, and then they become exhausted. Whereas the parallel CAR can be stimulated through many more cycles on average. And when you move this system in vivo, you can see that these PCAR T cells have efficacy in tumor xenograft models, which are refractory to conventional third generation linear fusions. The PCAR that I want to tell you most about really is our lead clinical candidate at Lucid Bio. And this is a parallel CAR which targets the NKG2D ligand family. NKG2D is a receptor found on NK cells, gamma delta T cells, and CD8s, and it is a surveillance receptor looking for evidence of stressed cells throughout the body. And a cell indicates its stressed state by upregulating NKG2D ligands. Most tumors are stressed cells and they will express these ligands. And indeed, if you take mice which are knocked out for NKG2D, they will develop tumors, spontaneous tumors, at a significantly higher frequency because they have lost this surveillance mechanism. So we thought this might be an interesting set of targets to go after with a CAR T cell based approach. Here you see some tumor restimulation experiments, and it's the same message as I delivered earlier with the CSF1R model. You can restimulate these PCAR T cells through many cycles on different tumor monolayers, which express the target. And the other attraction of targeting these ligands relates to the fact that they're not only expressed on the stressed tumor cells, but they're also expressed on the stressed stromal cells present in a tumor. What you see here is a 2D cell culture model where we have tumor cells, in this case pancreatic tumor cells, which form discrete islands within a sea of mesenchymal stromal cells. Now, the difficulty with stroma in these tumors is that stromal cells tend to elaborate chemokines, which naturally attract T cells to that location. And of course, when they're sitting in the stroma, what they're not doing is attacking the tumor. But that doesn't afford any defense to this system because the stromal cells also express NKG2D ligands. And so if you add these CAR T cells, they initially make a beeline for the stroma, which you can see they're now destroying. The tumor islands seem to coalesce, but this doesn't protect them because the CAR T cells next turn their attention to the tumor cells and they destroy those as well. And you can also model this in a 3D tumor spheroid system where you have a mixture of tumor cells flagged in green and stromal cells, which are tagged with a red fluorescent protein marker. If we add the CAR T cells, if we add the CAR T cells, you can see that it takes them a few hours to actually break down the outer barrier in this tight ball of cells. You can see that about now they are beginning to break into that tumor spheroid. And what they then do is they just infiltrate through the spheroid and destroy both the tumor cells and the stromal cells, which are present indiscriminately, more or less. And in fact, you can even see the end product of that with the naked eye. So here you can see 5 tumor spheroids, which have been co-cultivated with untransduced T cells. And at the end of the experiment, you can still see that those spheroids are intact. Whereas with a linear NKG2D CAR, again, the spheroids are more or less intact. With the parallel CAR, on the other hand, they're completely invisible. And if you deaggregate those spheroids and put them through a flow cytometer, you can see tumor and stroma in these 2 cases, but not with the PCAR. And this translates into significant efficacy in a number of different solid tumor xenograft models. Here we have pancreatic and mesothelioma xenografts. And here we have a PDX that we have developed in-house from a mesothelioma tumor, all regressing when they are treated with these PCAR engineered T cells. So my conclusions are very simple. When designing CARs, thinking about the signaling moieties, it's all about the location of these moieties. And I would like to stop at that point and thank all of the people in my academic group and also the core mechanics, also Lucid Bio, our spin-out company, who have generated all of the data that you have seen. I'll pay particular mention to Mark Davies, who developed the T4 CAR back in the day, a long time ago, and also Sophie Papa, who's a medical oncologist who has been working with me in the delivery of the phase 1 clinical trial. And I would be very happy to take any questions that may have arisen following the presentation.
So thanks, Dr. Maher, again for your nice presentation. We have a couple of questions coming through the chat. So the first one is a technical question from Jason Lee. They ask about how you transfect CAR T cells. Are they stably transfected, or this is a transient transfection?
Yeah, so in general, both in our kind of preclinical work and also in the clinical trial, we use a retroviral vector. So it's quite an old vector called SFG, which is very widely used. So it gives you a stably engineered gene product. So generally that's what we do. There is a significant move in the field at the moment actually towards the evaluation of non-viral alternatives. And I think that's probably going to be an expanding arena of activity in the not too distant future.
Thank you. And then another technical question is related to the way of the administration of CAR T cells. Are these given IV or you inject these intratumorally? So I think it's related to the first part of the talk.
Yeah, absolutely. So in the trial, we're administering the cells directly to the tumor. So what we do is we formulate these cells as a fresh product on the final day of manufacture, concentrate them down so they're at very high density, and then inject the cells, aiming to deliver to multiple viable points in the periphery of the target lesion. And when we do that, we have not seen any evidence of leakage of the CAR T cells into the peripheral blood as a consequence. There are only so many tumors you can play this trick with, of course, and ultimately we will need to think about systemic delivery, and we are working on strategies to make these CARs safe. One quick example, we have recently published a study with James Arnold at King's in which we converted this CAR to a hypoxia-regulated derivative. So the CAR is only expressed in the hypoxic tumor microenvironment.
Very nice. So another question coming through the to the chat is related to the side effect. So is this therapy like leading to development of any type of side effect, like generation of immunity or any type of like exacerbated inflammatory response?
Yeah. So in the context of T4, it's pretty safe, I think it's fair to say. We don't see any systemic toxicity. Generally, the patients develop fever on the evening of treatment. It's usually gone within 24 hours, sometimes 36 hours if you give the higher dose level. Also, the target lesion tends to become inflamed. Which I guess is a good sign, you might argue, because you're seeing the CAR T cells are becoming activated at that site. But that can become uncomfortable sometimes, and so you may need to give analgesics. In some of the patients, even corticosteroids have been given to damp down that reaction, but no systemic toxicity. So no evidence of cytokine release syndrome, no evidence of neurotoxicity, as is commonly seen with CD19 CAR T, and no dose limiting toxicities. So, one of the big fears we had starting out was what happens if these T cells spill out of the tumor and enter the bloodstream and end up in the liver or in the lungs? What kind of toxicity might we encounter? But we don't see that kind of leakage happening with this intratumoral delivery type approach.
Thank you. So maybe we can have another question, and then I will leave the other for like a private chat to stick with the program. So is a question related to the source of the organoids. So they're asking if these organoids are prepared from primary cells from patients or any other source.
So no, these are immortalized cell lines. So for example, we have a number of different mesenchymal stromal cell lines. For example, pancreatic stellate cells, which have been immortalized, and also the tumor cells are immortalized as well. So in those models, it's generally immortalized starting material.
Okay, thank you a lot. I think we have a few other questions, but we should go ahead with the program, and maybe no, we have time. They're telling me that we have time. So yeah, good. So yeah, I mean, then another question is, are T4 CAR T cell progressing to later clinical trials, or do you aim to preserve a central memory phenotype in your expanded CAR T cells?
Okay, so there's two questions in there. Yes is the answer to the first question. So we're just about to submit, I think it's our 11th substantial amendment in this clinical trial. That's one of the things you learn when you do these kind of trials. There's just no end to the paperwork that you end up being immersed in. But in any case, what we propose to do moving forward beyond the lymphodepletion cohort is to add in 3 doses of a PD-1 checkpoint inhibitor. So the first of which would be given before the CAR T cells are infused, and then 2 further doses at 4 and 8 weeks after the CAR T. And the idea there is, putting these CAR T cells into that hostile tumor microenvironment is clearly going to favor their exhaustion. Can you rescue any of that by blocking PD-1 on the CAR T cells themselves? So that's the direction of travel. Regarding state of differentiation, that's a very difficult issue, to be honest. And we have tried to make life as simple for ourselves as possible in this, our first clinical trial of CAR T. So we're using whole blood, PBMCs, and we're not selecting for any kind of subpopulation in any stage of the process. The price you pay for that is you get a lot of variability in your end product. Sometimes they're predominantly CD4, sometimes they're predominantly CD8, and you see every shade of gray in the middle as well. And that also applies to the state of differentiation of the cells as well. I agree, however, with the questioner. Again, this is a direction of travel that is very likely to be pursued, where people identify subpopulations, particularly less differentiated subpopulations, as starting material, and also potentially adding in pharmaceuticals into the manufacturing process, such as PI3 kinase inhibitors, for example, to try to keep these cells in as a young a state, as it were, during manufacture, because we know that fitness of these cells is a key metric in achieving success.
Very good. And then a question related to the PCARs. So basically, how these PCARs compare with first, second, and third line generation in terms of remaining active and if they are able to signal every time when they are stimulated, or eventually if they get exhausted?
They do eventually get exhausted. They're not kind of immortalized cells, as it were. So if you stimulate them enough, they will eventually become exhausted. But they are more persistent than the traditional linear fusion. So they do outperform the competition, as it were. And we think the reason for that is because you're benefiting from the dual co-stimulation of CD28 and 4-1BB. 28 is a kind of a the type of co-stimulation you get a rapid expansion, rapid cytokine production, a bit like the tortoise and the hare, as it were, whereas 4-1BB, on the other hand, is a more of a slow burner. And there are advantages to both, in a sense. And from my perspective, if you can capitalize upon both types of co-stimulation, you have the potential to have a better product at the end of the day.
Thanks. Then another question on NKG2D. So basically they're asking, since this receptor is expressed also by human CD8 positive T cells, why do you think that this receptor would do the job only for CAR T cells and not for endogenous?
When it's already there in nature.
Yes.
And the point is, in nature, in the human system, NKG2D only signals through an adapter molecule called DAP10, which provides a co-stimulatory signal. What we're doing with the adapter CAR system is we're delivering an activating signal as well as a co-stimulatory signal. And indeed, we're not the only ones doing this. There are a number of other people prosecuting this target with various CAR T approaches, such as Celiad Oncology, for example. So that's the key difference. With the CAR system, you can also deliver, whatever types of signals you believe to be important. In nature, in the human setting, NKG2D only provides a co-stimulatory signal within the context of T cells.
Another question related with side effects. So have you checked allergy development in the patient treated with CAR T cells?
Interesting question. I mean, I'm not really sure what checked means. We don't exclude people who are atopic, because if we did, that would exclude 1/3 of the population, which would be, and when you're recruiting to these trials, you realize you want the minimum number of barriers, because recruitment is always an issue in these kinds of clinical trials. Allergy to CAR T, to my knowledge, has only ever been reported in one patient. It was a patient treated at the University of Pennsylvania, and that patient had an anaphylactic reaction following CAR T administration, which was believed to be IgE mediated, although they never actually identified an IgE antibody with reactivity to the CAR. So it can happen, but experience to date suggests that significant allergy to CAR T is an unusual phenomenon.
Okay, so I think we can move to the next talk. So for people who just connected, Matthias Bozza is an expert in the development of autonomously non-integrating DNA vectors, and he works at the German Cancer Research Center in Heidelberg in the laboratory of Dr. Richard Harbottle. And today he will deliver a talk about non-viral autonomously replicating DNA nanovectors for T-cell engineering. So please, Matthias.
Yes, apologies for the technical problems. I hope now we are all sorted. Yes, so as the keynote speaker introduced, there are several ways to introduce a gene of interest into T cells. If we can move to the next slide. We can use either viral systems, which are currently probably the state of the art. We can use what I call a non-viral dual component system that are like CRISPR-Cas or transposon systems, or we can use plasmids. And my presentation will be focused on the usage of plasmids for the introduction of genes into T cells. Can we move to the next slide, please? What the obvious question is when we started to work with plasmid is, why would we use plasmid DNA when there are several other technologies available? And there are a number of obvious answers. And the first is that when we introduce plasmids into cells, they don't damage or kill the cells after they have been delivered. They remain episomal, and therefore they reduce the risk of integrative mutagenesis. And by non-integrating, they also reduce the possibility of cis or trans suppression or activation of endogenous genes. If you click one slide ahead, we do see, however, the problem that when we introduce plasmid into rapidly dividing cells, the more the cells divide, the more we lose the expression of the transgene due to a simple factor of plasmid dilution into the population. And on the right side, there's a clear example of a GFP follow-up of a plasmid delivered to rapidly dividing cells that were very bright 24 hours post-deliver, but that became particularly dim 15 days post. So could we move to the next slide, please? So we started to work on these concepts, and as soon as we started, we kind of withdrew a wish list, and we said, how do we want the plasmid? How do we want the plasmid to be? And firstly, we decided to have a technology that was fully non-viral, that could be episomally maintained in the target cells, that has an unlimited cargo capacity to increase the number of genes and the size of the genes that we can deliver, that can be used for several applications, such as in vivo, in vitro, or ex vivo, and that provide long-term gene expression. And I think that the long-term gene expression is probably currently the bottleneck, the most relevant and most difficult point of this list to achieve. So if we move to the next slide, we see that we have been developed plasmid based on scaffold matrix attachment regions. These regions, known as SMARs, are widely spread in the nuclei of every species. And their natural function is to tether the DNA to the nuclear lamina by creating loops. And those loops are not only involved in the generation of the chromatin structure, but those loops are also characterized by a highly transcription activity. So if we go one step ahead, when we moved these sequences into plasmid, we found out that those are sufficient also to maintain the plasmid tethered to the nuclear lamina, which means that these vectors, once they are introduced into the cells, they are replicated and they are maintained as if they are an endogenous part of the nucleus. As a consequence, once these vectors are delivered into cells, they can stick there, they can be replicated, and they can be segregated into the daughter cell, resulting to a long-term persistent and gene expression. So if we move to the next point, we developed these vectors and we thought about them as multi-factor systems, where in the plasmids, the sequences are not only physically, but also functionally in relation to each other. So the vectors are, as I mentioned, are multi-components, and the four main features that they present are summarized on the table on the right side that will appear as soon as we click. Exactly. So the first features is the bacterial backbone that is necessary for the plasmid production in bacterial, but it actually also gives the overall immunogenicity of the vectors into human cells. The second features are the presence of cis-acting sequences. These sequences are important to shield the vector, for example, from epigenetic silencing events or to enhance the transcription. Third is the introduction of 5' and 3' UTR that stabilize the mRNA. And the fourth is obviously the presence of an SMARs. So now if we go to the next point, here is briefly summarized what the technology that we use to produce this plasmid that is not based on antibiotics such as kanamycin or ampicillins. So we rely for the production of these SMAR plasmid on the nanoplasmid technology developed by Nature Technology. So the plasmid production and selection is mediated instead of antibiotics from an interfering RNA. And without going very deep into the details, the idea behind is that if the cells produce an enzyme that is toxic in the presence of the sucrose, when the plasmid is present into this bacteria, they produce an interfering RNA that can induce the knockdown of this toxic enzyme that allows the selection of sucrose. And if we click exactly, and these vectors are easily produced in bacteria, as I mentioned, as a normal vector, and the scale up was also successfully achieved, and they have a particularly high DNA yield that peaks about 2 grams per liter during manufacturing. So now there are a number of advantages by using what we call N-smart vectors, these nanosmart vectors. As I mentioned, they are fully non-viral based on mammalian DNA, episomally maintained. They provide persistent expression. They are produced using an antibiotic-free selection system that we just saw. They have an unlimited capacity, and they can replicate, more importantly, only in a specific strain of E. coli, reducing the possibility of, or actually minimizing the possibility of replication into the bacterial host when used for human application. So now going to the next, when we tested here as an example, these vectors in rapidly dividing cells, such as GIRCATs, as you can say, we can maintain the vectors up to, in this case, was a 150 days follow-up, where we didn't lose, where we didn't see a significant shift to the shift towards a negative cells when we looked for GFP expression. We then recently published a paper where we showed this vector development that actually was a long ride. It was about, I think, almost a decade of development for making vectors efficiently, to make this vector efficient for expressing CARs or TCRs in T cells. We then compared this vector to the current clinically approved state-of-the-art lentivirus in vitro and in vivo killing. In vivo killing assays, and we also developed a novel manufacturing protocol. If we go to the next slide, by using these nanosmart vectors, after along the developmental phase, we can now achieve very high and very efficient delivery to T cells by using electroporation, independently from the genetic cargo, being it a CAR, as you can see on the left side of the slide, or being it TCRs. So this is variability that was measured 1 day post-delivery, that has a tendency to increase in the days after the delivery. What was important for us was to find the nice balance between efficacy of delivery and viability of the cells post-electroporation. We then move to the next slide, and we then decided to compare side by side the activity of T cells that were engineered using N-smart technology and cells that were made with a lentivirus. As you can see, the efficacy of delivery, we normalized the efficacy of delivery in order to have a similar efficacy of around 80%. And then we run an in vitro real-time killing assay with an effector to target ratio of 1:1, where for both cell lines expressing the target, one's at low level, such as HT29, and one at the high level, such as MCF7. In both cases, we could demonstrate that cells made with N-smart were more efficient in recognizing and killing their targets. So we then investigated the reason of this better efficacy. And if we move to the next slide, we are identifying by single-cell RNA-seq that when we deliver the CAR, in this case with an N-smart vector, we found about 60 genes that are perturbed, in which their transcription is perturbed. And this is a single-cell RNA-seq analysis. While when we do the same experiment using a lentivirus, harboring the expression of the same CAR, we find almost almost double of the genes that appeared perturbed, and that most of these genes are involved in a pathway that are involved in the fighting of they're involved in the interferon beta responsive pathway, which are pointing towards a hypothesis that the cells are actually involved in the fighting of the viral infections. So then when we compared, if we go to the next slide, when we then compared again the cells to lentiviral transduced cells, and we tried to find out how many now of lentiviral transduced T cells are necessary to match the functionality of one transfected cells with N-smart. If we do a double click, then we can go another one. Okay, we found out that 4 transduced T cells are necessary to match the functionality of 1 transfected T cells, which actually underpins the idea that if the cells are involved in fighting a viral infection, they have less energy to fight against the tumor. If we go to the next slide, We then evaluated the efficacy of targeting tumor cells in xenograft model, and also in this experiment we demonstrate the cells that were made with our non-viral approach significantly delayed tumor growth compared to cells made with the lentivirus that actually induce also a longer mice survival. These models, although it's not a perfect models, because what we faced in the cells that escaped the therapy is that they lost the antigen expression, and therefore they were not targetable anymore from the cells. However, and if we go to the next panel, however, what was important for us to see was that the cells were engrafting in all the significant organs, what we thought were significant organs, such as the tumor, so we could detect cells TILs in the treated group, in the spleens, in the circulating blood. And more importantly, and this is the panel on the bottom left, the cells that we could retrieve from the splenocyte of the treated mice were still able to recognize and kill the tumor in vitro after being for 40 plus days into the animal. And on top of this, when we characterize the DNA, the status of the DNA in these cells, we could find by rolling circular amplification that the plasmids introduced by electroporation, so the SMART plasmid, were maintained in its episomal form. And for the non-tactical, the rolling circular amplification is an assay that is specifically designed to amplify only circular DNA. So now, if we go to the next panel, we had to with the aim of applying these vectors into the clinic, we took advantage of the protocol developed for the manufacturing of human CD3 with the lentivirus, and we slightly changed it to make it compatible to our system. So here, as you can see, and you're probably familiar with, the protocol starts with the manufacturing of the virus, which is already quite a challenge considering the global shortage of manufacturing capacity. And then it needs the sample, then it needs the leukapheresis from the patient. The cells are activated, transduced, and further expand, a process that takes about 15 days. If we go to the next panel, this is the protocol that we have been optimizing. The protocols also, in our case, starts with the leukapheresis. We do positive selection of the CD3 cells with the automated cell process system provided by Meltini. So this is the Prodigy. And then we activate the cells, the CD3 cells, with the CD3 CD28 agonist and cytokines for 3 days before we deliver the plasmid using flow electroporation system provided by MaxCyte. This is an expert device provided by MaxCyte. And we reintroduce the cells into the Prodigy for other 3 days before we harvest the product. And as you can see, in both cases, delivering CARs or TCRs, we could achieve really high level of positive cells, maintaining the viability high. A advantage over existing technology using the SMART vector is that by having such a high yield during productions, which is about 2 grams per liter, with only 1 liter, we can produce DNA that's for about 1,300 patients, which in the end makes the biggest benefit for the patient that can access this therapy much faster. Now going to the conclusions. I think that I hope that I have convinced you that this class of vectors has a unique advantage that combines very low immunogenicity together with mitotic stability, that is produced at a high yield in a normal bacteria without additional post-processing. And you might be familiar with the post-processing purification steps that are needed with other minicircular technologies. They provide robust transgene expression, in this case in T cells, but we tested in many other cell systems, and it was consistently reproducible. And when it comes to clinical application, is comparable to the current state-of-the-art lentiviruses, but it has advantage of a shorter manufacturing time. So to conclude, I'd like to thank the people in our lab, Dr. Harbottle, who's my supervisor, and our collaborators, especially Dr. Green and Dr. Correa, who were really helping moving this project forward. And I'll thank you for your attention.
Thank you, Matthias. So we have a few questions coming through the chat. So the first one is related to the platform you used. So if you can can elaborate more on the and comment more on the platform you have used to generate these new CAR T cells. I think this question was asked before you were showing your last slide on the platform with the Miltenyi system.
Yeah. Yes. So for the clinical application, we used both. So we combined the use of the Miltenyi Prodigy system for this is a automated cell sorting and cell culturing device, and the vector is delivered, as I mentioned, with the Expert MaxCyte flow electroporation device.
And then a general question: do you think that this kind of technology will become a golden standard for CAR T cell research and clinical trials?
I hope so. I honestly do think that non-viral in general is going to be the golden standard. I don't think there will be much space for viral manufacturing in the future. If these or transposons or CRISPR is going to win the race, it's hard to say. I'm sure that CRISPR and transposon might be a little ahead, but we are working out to catch up.
Oh, sorry. Probably I wasn't that clear. So when I read this question, the question was related to the single cell platform you used at the beginning. You show some data on single cell sequencing, RNA sequencing platform, and what type of single cell technology have you used?
That's a multi-omic approach provided by Proteona as a company, as a service company in Singapore, that can. That basically analyzes the transcriptome, so the single cell RNA and the proteome-seq, single cell RNA and proteomic on the same sample.
Thank you. And actually, on the top of this question, so you showed these two transcription profile from the CAR T generated with the viral, so the lentiviral one, and your new innovative system. And in the one generated with the viral, with the lentivirus, you saw this strong upregulation of type 1 interferon-related genes. So I wonder whether this can be due to the fact that you are actually using a virus and your T cells can somehow, let's say, induce some gene just because they are exposed to this virus. And in your system that is viral free, you don't have this upregulation of these type 1 interferon related genes.
Yeah, no, that's clear. I mean, that's becoming clearer and clearer that by the T cells as part of the immune cells, they are designed by nature to react against foreign entities, and by using the virus, it clearly knows that it is kind of under attack and under infection. While if you use a non-viral, well, that was the 10 years of development behind to make a plasmid invisible, that the cells doesn't think, hey, I am infected by a plasmid.
Yeah, paradoxically, it seems like an innate response that it shouldn't be the case for T cells, but apparently they start producing interferon-related genes after being exposed to lentiviral. And then a technical question related to the way of culturing your CAR T cells. So have these been cultured in presence of IL-7, IL-15 to induce this naive central memory T cell phenotype?
Yes, so exactly. Culture is with CD3, CD28 agonists, and IL-7, IL-15 to encourage naive central memories phenotype retention, as it was shown also in recent publication. But the CD3, CD28 is washed out from the culture after 48, 72 hours. So after the first expansions, activation phase, so after the first 3 days, the CD3 and 28 are washed and they are further cultured only in the presence of IL-7, 15.
And then there is a last question that I have skipped, sorry. So can the transfected T cells selected as a memory T cells? can be selected as.
Yes, I guess if you sort for specific markers, you can. So we don't do it. We just use the whole population. But I think why not? It should be fairly easy to do.
Our last talk will be given by Dr. John Liu. Dr. John Liu is currently the president and the CEO of Habesal Corporation, that is focusing on the development of clinical translation of regenerative medicine and cell therapy technology. In particular, his work focused on iPS CAR NK cells for the treatment of cancer, autoimmune diseases, and also viral infections. Before establishing Habesal, he was the Senior Director of Research at Advanced Cell Technology, Okta Therapeutics, and we are very pleased to have Dr. Liu, that is an expert in stem cell biology and regenerative medicine. He has more than 20 years of experience working with human embryonic stem cells, induced pluripotent stem cells, and their derivatives. Also, and this is quite remarkable, he is the inventor of more than 20 patents and in this stem cell field. So today we will be listening about CAR NK cells, from engineered pluripotent stem cells, off-the-shelf therapeutics for all patients. So please, Dr. Liu, whenever you're ready.
Thank you. Can you hear me?
Yes.
Yeah. Thank you very much. Thank you for the nice introduction. Thank you for the organizer and Proteintech giving me the opportunity to share you what we are doing in Habesal. So let me start with very nice introduction by our first keynote speaker about the CAR T cells. So what I characterize the CAR as a GPS for the missile for effector lymphocyte, because the first part of is the GPS, the intracellular is like a missile. But mostly right now, I think CAR T cell therapy usually use autologous PBMC to isolate, I think, T cells and expand them and engineer with the CAR, and then expand, then re-infuse into the patient. So there are tons of progress during the past 10 years, and specifically recently, I think there are 2 more CAR T product was approved. And right now, probably this is 2019, right now, 2021, probably more than 1,000 CAR T trial, I think, in the worldwide. This progress also has met a lot of problems, a lot of things. Most of them can deal with biological challenges, I think, by design, the cars, a lot of other things. But what I'm talking about majorly today is about the manufacturing challenge, which is, I believe, is the major obstacle as the therapeutics. Why? Because since, as we know, most CAR T cell therapy right now using autologous product. So as we know, the patient, late stage patient, about 30 to 50% of patients with late stage patient, they have exhausted T cells. That means their T cell is not good enough for therapy. So secondly, it's a high cost. But each one right now, the minimum would cost $500,000, and ordinary people were not going to be able to, I think, pay it. So secondly is a limited of availability, as I said, because a lot of patients, and they don't have good T cell for their CAR T cells. Most important is the heterogeneity for different patients. That means your efficacy cannot be standard or cannot be achieved even using the same manufacturer. So the other major problem is the manufacturing time delay, because you need at least 2 to 3 weeks to get from the blood to the CAR T engineering and expansion, then infusion to your to the bodies. So, so what is the solution? So I have some summary with other people's input. I think what is the ideal immunotherapy product, cell therapy. So first one, the cell therapy product should be targeted. That means the maximum on a target effect and minimum on off-target toxicity. And this can be achieved by the CARs as introduced into the cells. A second one is standardized, which every patient you get the same treatment or same cells. That means they are getting the same product. And the third one is off the shelf. That's very critical because you don't need to produce the product for each patient, and the cells are ready and stored. When the patient come here, when they need any kind of CAR cells, you can just infuse to them, give it to them. And the last one is centralized manufacture, because this is very, very critical, because right now most CAR T therapies and the manufacturer is in the hospital, and each hospital is different. Can we make a centrally manufactured under GMP condition as distributed to worldwide to any patient in the world? So the ultimate goal is to make a cell as easy to handle, use as a conventional drug. And can we do this? So actually, CAR T cell, NK cell is the perfect fit for cellular immunotherapy. Why? Because NK cells is cytotoxic lymphocyte. They kill target without HLA restriction and without prior centralization. And secondly, they kill virus, infected cell, and tumor cells rapidly. And if you plot the CAR, they are more specific. And their cytotoxic is regulated by the balance between activating and inhibitor receptor. That means they are not going to kill normal cells. And most important things, this can be allogenic and without evidence of GVHD, which is different from CAR T cell. CAR T cell, or if you're using allogeneic T cells, they can generate GVHD if you didn't do gene modification. The most important thing is, I think actually NK cell as preferentially kill cancer stem cell, which is divided slowly and has very low HLA expression. So by NK cell can kill virus infected cell and tumor cell by different ways. However, if you put the CAR engineer into the cells, they can more specifically targeting a specific target. I think that's more specific. So the problem right now and in our body, the NK cell not so much. How can we get it? So one thing is hemopoietic differentiation from pluripotent stem cells. I think we mimic the in vitro hemopoiesis, so we can generate from myeloid cell, lymphoid cells. However, during the process of development in stem cell differentiation, the myeloid lineage is very easy to achieve. I think we have achieved the red blood cells, megakaryocyte, and platelet, and macrophage, and neutrophil, a lot of things. However, for the lymphocyte, it's much more difficult because this is a definitive, I think, lineage. So in the NK cell and development in the body, basically they are in the bone marrow, then migrate into secondary lymph tissues. From there, they develop into specifically natural cells. The problem, can we mimic the secondary lymph tissues in culture? So by doing so, and we develop 3D culture system, try to mimic the in vivo secondary lymph organ tissues. So regularly, I think every culture, ESL, RPSL, using 2D culture the dish or flask, and they culture them. And during the past several years, we have developed a 3D culture system, and we culture them, the RPSL or ESL, as a sphere. And by then, we push them, differentiate them. So in vitro or in vivo, the hemopoiesis has several waves. And initially, I think for differentiation, they have a hemogenic intracellular population. Then the first wave, most of them are megakaryocyte and erythrocyte. And the second one usually is a myeloprogenitor cell. These two are very easy. How can we get to the third wave, which is lymphocyte specific? So we try to do this in mimic as in the immune system to get the second, the last one is NK specific differentiation. So by doing these things, and we have developed a process can block all this differentiation and only leave the NK cell differentiation. I think I will show you how we did these things. So by doing these things, first we culture ESL, iPSL, then we bend this ESL, iPSL in a spheroid condition. I think by doing these things, then we induce them to a homogenic endothelium differentiation process. That we have about usually take a week or two and can get about 60 to 70%, I think, expression of the CD31 positive cells. Then after that, we switch medium, push them to hematopoietic progenitor cell for another week. By these things, then we totally switch them into NK cell differentiation conditions. By doing this, we see this field right now, they are mimic the secondary lymphoid tissues. I think these cells are differentiated into NK cells. These cells can release into the medium, and you can see the empty cavity. And these cells, when we release from them, they are very pure. Now, we don't need to further purify them. And how do we do these things? Here is 300 ml bioreactor. This is all the spheroid. By doing this, we can generate about 10 to the 10 to the 9, I think, primary like NK cells, I think, from these bioreactors. So these bioreactors conditions are defined and very scalable conditions. I think these we can generate homogeneous and highly pure preparation of NK cells. So when we get cells, what people ask, are these real NK cells? I think these things tell you, and they are this before any purification. NK cells are very different, and from other, I think, hematopoietic cells specifically for the T cell, B cells. Their morphology is very distinctive. I think this is without any purification. Then we run a FACS analysis to check, do they express NK cell markers, and they do, and about 95% of them express CD56, but they do not express TCR or CD3 markers, and they do not express B cell markers, CD19. However, they do express a lot of NKG2D, I think about 95%, NK, P45, P46, and all other markers. What also express the inhibitory receptor. If they do not express the inhibitory receptor, they are going to kill all normal cells. One more important thing here, our cell, I think 60 to 80% express CD8, which is not just a T cell marker, it's the effector cell markers. Actually, this cell may be more cytotoxic. To further confirm our cell are typical NK cell, we have a collaboration with Dr. Chen at MIT. We send him 3 sets of 3 lot of NK cell we prepared, and we do the single RNA sequence analysis and compare with 2 database in the internet. So basic analysis, I think they provide all this analysis, and they told us, and we compare them. They are all iPS CD50 positive cells are typical NK cells. So right now we have the NK cell, and we're now, so can they kill cancer cells, but not normal cells? I think first we're using K562 cancer cell, which is a typical NK cell target. And also we use normal PBMC, which is allogenic, and from normal, I think, people. Then we see where they can kill them. So from here, you can see these cells are very actively tracing these large K562 cancer cells. And after they get them, they are going to kill them. The same way, if you put normal, I think PBMC, they still tracing them, but they are not to kill them. So they just trace them to see they are not to kill them. You can see cell in the pop up, but here they just trace them. They are not going to kill. So let me then we do the effect analysis to confirm the result for the movie. you can see, and the same thing, you can see the analysis before mixing, and after 2 to 3 hours, you can see K562, most of them are killed, about 80% of them. And same thing for the PBMC, they don't kill at all. So we have also tested several other cancers like AML, pancreatic, ovarian, colorectal, and lung cancer. They did the same thing. I'm going to show you one experiment. So we did some ovarian cancer. I think we culture them on a dish, and then we add it with our cells. I think that's overnight culture overnight because they are not as sensitive as the K562. About 40 to 50% of the ovarian cancer were killed by our NK cell by overnight cultures. That confirm they are going to be able to kill a lot of cancer cells. So next step we want to see, because since the NK cell is not just kill cancer, they are the first defender for viral infection, specifically for AIDS patients. Why? Because AIDS patients, late stage AIDS patients, is very, very difficult to treatment because CAR T cell is almost impossible because NK cell, HIV virus primarily infect CAR T cells. So NK cell can directly recognize and kill HIV infected target cells. I'm going to show you some data later. So the KRR on NK cell are able to inhibit HIV replication and prevent HIV progression. Most importantly, NK cell will not be infected by HIV virus because this virus primarily infects CD4 positive T cells. So I'm going to show you our NK cell can recognize and kill virus infected cells by HIV, dengue virus, influenza A, and Zika. We try to do a SARS-CoV-2, but we cannot get the samples. So in collaboration with Dr. McDougall in University of Massachusetts Medical School, we have a normal, I think, a primary epithelial cells, and then we infect it with different virus, and then we culture it with our NK cells. You can see with no virus infection, the NK cell will not kill them, but influenza A, Zika, and dengue virus, HIV virus, after culture about 6 hours, they kill almost all of the infected cells. That prove our NK cell can be used for prevention or treatment for virus infections. Another one we are still planning to do is for autoimmune disease, because as we know, autoimmune disease was caused by either B cell or T cells. I think by chemical autoantibody receptors, we can target specifically B cell which they secrete autoantibody. There are several CAR T cell clinical trial and do these things. And same thing, we can use NK cell express a PD-L1 to target specifically follicular T cells for the control of lupus. So by doing these things, we can use it as a CAR NK cell to treat autoimmune disease. I think as a title, I think the CAR NK cell from engineered PSC can be used as off-the-shelf therapeutic for all patients. How do you do this? So have a strategy. First, you have a iPS cell line or ES cell line. You can gene editing them at an early stage, then select the stably gene edited cell line from here, and this gene editing can be used to enhance, I think, NK cell functions, and you can introduce different CARs. And first, you can use a CAR targeting tumor antigens and targeting viral antigens or the autoimmune antigens. By doing this, you have generate a multiple CAR master cell bank. By doing this, you differentiate them using our bioreactor into a CAR NK cell, and this NK cell can store in liquid nitrogen. And when our patients, the patient come to the hospital, you just deliver them and store them and deliver to them. And same thing for all other things. By doing this, you have a master cell bank and can differentiate and into a different specific CAR NK cell for all patients. And so as a proof of concept, we have a CAR and with GLP linked together. You can see we can select very stable clones of the iPS cell, and they culture them as field and differentiate the NK cell. And the all of them express about 94% of them express the CARs. And that's very different from all CAR NK CAR T cell therapy because most of them get about 50 to 60%. That's good enough. So what are our advantage of our platform? So this is a platform technology, the unlimited CAR NK cell type targeting multiple indications. I think our patent 3D platform is scalable and very consistent with different cell line and CAR insertion and gene editing, and can generate homogeneous functional NK cells. And also the most important is the cost effective. So with the build-up of master cell bank, and we can virtually produce unlimited iPS CAR NK cells. And this production will define CAR cells with a uniform, almost 100% of them contain a CAR, and with very consistent pharmacological profiles. They are off-shelf, and we can deliver to all worldwide to different patients. So I just have a simple comparison with the current use of CAR T cell, donor CAR NK cell, and PSC CAR cell. Most of them they are very similar. However, there are two distinct, I think, properties compared to PSC derived CAR NK cells. One is uniform, and these cells you just select permanent clones. When you generate cells, they are almost the same. And here is a very random variable. As a specific for NK cell, virus infection will cause a lot of cell death. Secondly, most important things, and we can generate a cell bank for multiple CAR targets for all dominant for the donor cell they cannot. So these are the two most important advantages. So I think right now there are a lot of CAR NK cell therapies, iPS or ES cell development. Most of them they use 2D culture and with 2 or 3 feeder cells. And for our purpose, we only need one bioreactor from beginning to iPS or ES cell expansion, differentiation, and finally the collection of NK cell. And they are multi-step, I think, different cell feeder cells, and a lot of work. So right now in the therapeutic space, I think donor-derived NK cell, and they are all, I think, very crowded. I think they expanded with the cancer cell as a feeder cell, and the feeder cell has to be similar, treated as similar as the therapeutic cell because you have the GMP compliance. I think in the iPS or ES derived NK cell, the space is not so crowded, but there are at least right now about 6 to 7 or 8 companies are working on it. But most of them are still using 2D culture. They still need the cancer cell as a feeder cell to print them. For us, I think we are the only company right now in the world can use the bioreactor without any feeder cell to generate primary like NK cells. So that's our advantage. So that's all. I think if anybody you are interested, join us, and we are expanding. And please send your CV and to us. We are going to contact you. Thank you.
Thanks very much, Dr. Lu, for this very interesting talk and this very powerful technology. So we have a couple of questions coming from the Q&A chats. So the first one is asking if your fresh NK cells prepared from iPSC are already able to kill cancer cells, why you need to make them as a CAR NK cell?
That's a very, very good question. So I think, NK cell have a lot of active receptors, like NKG2D. I think they can recognize both tumor and, I think, infected cells, antigens. However, as the first keynote speaker said, they may not be that powerful compared to have contrast CAR structure. That way can give them more, I think, signals. That way the killing will be more powerful. Secondly, I think NK cell receptor record multiple, cancer cell antigens. And if you have a CAR, you can specifically target and concentrate on a specific tumor type you want to kill.
Thank you. And the next question is related to the possibility for CAR NK cells to become exhausted. Is this happening?
Exhausted?
So if these CAR NK cells get exhausted,
they do. So I think one thing is NK cell. I think right now that's a major problem right now for the donor derived and 2D. They need tons of expansion. They're using feeder cell to expand the cells. I think if you're going to check all the clinical study, and they claim they can expand 1,000 or 10,000 times, but most of them treat patients. Each donor only treat one patient. I believe that may be the limitation, because if you expand too much, the cell may not be good enough. For iPS CAR NK cell, we are not expanding NK cell as I show. We are expanding the iPS cells. We generate primarily like NK cells. For us, we don't have the problem. For a lot of donor-derived cell, NK cell therapy company, they may have, yes.
And the last question is, have you profiled the secretome of these CAR NK cells? Are they able to produce different cytokines like interferon gamma?
We did think after they activated, they express, I think, secrete all the similar, I think, NK cell activation cytokines like interferon gamma and a lot of things. They do. We have a lot of comprehensive analysis on these things.
Thank you. Thank you very much again.
Thank you.
And now I think there is Rebecca.
Thank you so much, Eduardo, for leading this really great event. And I just really want to thank all the speakers for taking their time to speak at our event. I thoroughly enjoyed it. I hope you all did too. And just before we go, I'd like to introduce Fiona, who's our protein sales manager. And she's going to quickly talk to you about our humankind protein range.
Great. So just quickly, I know we're getting short for time now. It's been really interesting listening to the presentations this afternoon, so thank you very much. Learned a lot. I'm just going to introduce the Proteintech range of humankind. That is a range of recombinant proteins, the chemokines, cytokines, growth factors, and interleukins. And we also make recombinant human serum albumin, which is particularly important when you're furthering your research to clinical and looking at removing all animal components. Humankind, the humankind advantage is really that all humankind products are expressed in HEC293 systems. Other recombinant proteins are expressed in E. coli or CHO systems, perhaps. So we have native human conformation, that the maturation and folding, the native human post-translational modifications, PTMs, is slightly easier for me to say, and the glycosylation is particularly in the heavily glycosylated proteins can lead to a higher biological activity and stability. So the real difference is that these are all human proteins that we manufacture. We're not manufacturing, for example, mouse proteins. They're all animal component-free, xeno-free, endotoxin-free, tag-free, and carrier-free, which becomes even more important when your research gets to that stage of being a preclinical and clinical product. From listening today, we also have a very relevant range for research purposes, which is the Nutrakine range of neutralizing antibodies for use in research, and we use the humankind proteins for the immunogens. So these can be really useful, particularly listening to some of the researchers' talks today. They're particularly relevant. So as I said, it was a quick whistle stop tour of humankind. please refer any questions over to me on Fiona@PTG Lab, and you can also Fiona Bello on Twitter if you or LinkedIn if you want to reach me there. And I'll pass back to Rebecca.
Okay, thanks, Fiona. Yeah, so that's it from us. Thank you so much for attending. We really hoped that you enjoyed it. And yeah, like I said, because we're lacking this networking that we normally get with conferences, so please keep the conversation going. Connect with the speakers on LinkedIn and Twitter. And yeah, thank you so much for attending, and I hope you have a good rest of your day.